Well, welcome back, everybody. The next lectures that we're going to be going through, the next three, are profiles of three different pathogens that I think are very important in public health and in your personal health. These are things that are covered in the textbook. They're in the different body systems, diseases that affect the body systems sections. So you can find information in there, but I will also be providing you with other sources of information at the end of the lecture. And what I'm presenting to you is a little bit of the history of the disease, what the disease causes, and also what causes this disease. The first one we're talking about today is HIV and AIDS. HIV stands for Human Immunodeficiency Virus. So as you can kind of tell by the name, it's caused by a virus. The type of virus that causes this disease is a retrovirus. So that means that it's an RNA virus, and it has to be converted to DNA first before it can be replicated. HIV, if left untreated, eventually causes acquired immunodeficiency syndrome, which is AIDS. AIDS is characterized by general loss of immune function. So it suppresses the immune system. Sudden weight loss, swollen lymph nodes, pneumonia caused by other infectious diseases, and Kaposi's sarcoma, which is a type of cancer. As I mentioned, HIV is caused by a virus. It's a retrovirus, and it's in the genus lentivirus. This virus is enveloped, so it has a membrane surrounding it. Inside, it contains reverse transcriptase, again because it's a retrovirus. So what that does is it takes the RNA that is in the virus itself and turns it into double-stranded DNA. One of the dangerous things about retroviruses is that these viral genes, once they're turned back into DNA, can then be integrated into the host genome. That means that viral DNA can insert itself into that host DNA. HIV comes in a couple of different versions, HIV-1 and HIV-2. HIV-1 is the most dominant form in the world. HIV-2 is found in parts of the world, but mostly in West Africa. There are even subtypes within HIV-1 and HIV-2 that exist as well. This is what the HIV virus looks like. I said it's a retrovirus, and it is enveloped. So on the inside here you have your RNA, and followed by the capsid, and it's an icosahedral capsid, and then it has the envelope on the outside, and you can see all these spikes that help it to bind to receptors on the host. Now specifically what HIV is binding to in the host are CD receptors that we find on T cells. So this is going back to our talk on acquired or specific immunity. T cells, like the helper T cells, have CD4 receptors. Those receptors are used by HIV spikes to bind to the host cells and enter them. The first step in HIV infection, again, is that binding. So the spikes on the outside of the HIV virus bind to the CD4 receptors. Again, these are T cells, helper T cells that we're talking about here. When that happens, the virus then gets inside of the host. The RNA is released, and also the reverse transcriptase. The reverse transcriptase takes the RNA from the virus, turns it into double-stranded DNA. That's right here, complete double-stranded DNA, that can then be inserted into the host DNA. After a latent period, so a period where we don't see any symptoms, the DNA is just resting, sitting in that host genome. Okay, this latent period can last for years, maybe even a decade. So after that latent period, something triggers the DNA to start to be transcribed again. So the DNA is transcribed into mRNA. That causes the mRNA from the virus to start to be produced. Now, as we know, retroviruses are RNA viruses. So once that mRNA is produced, that is the genetic material for the virus. That mRNA is then copied. Some of the mRNA is translated into proteins that make up the capsid of the virus, proteins that make up the reverse transcriptase. Once all of those components are produced, then the viruses are starting to assemble. So this is a virus that's starting to assemble here. We've got the capsid inside. We're going to have the nucleic acid put inside the reverse transcriptase. Then the last part is budding. Budding is where the virus particles are complete and they're released from the host cell. As you can see here, that's how the envelope gets on the virus. It takes part of the host cell membrane with it. Signs and symptoms of HIV infection include, with the initial infection, fatigue, diarrhea, weight loss, and maybe some neurological changes. So nothing really all that striking. But a chronic infection, a long-term infection, has more striking and unique signs and symptoms to it. Again, you have the weight loss, diarrhea, and poor nutrient absorption. But people with chronic HIV can have fever that lasts a long time, sore throat, even night sweats. They eventually start to have hormone imbalances, metabolic disturbances, and their immune systems start to wane. They'll get infections that they normally wouldn't have. Those are the opportunistic infections. Some really rare cases even cause lesions. So there's strange growths or abnormalities on the brain, meninges, spinal column, and even peripheral nerves. Those are nerves that you find in the limbs, like the arms and the legs. All of these symptoms go along with the course of the disease. Now, in HIV, again, there's an initial infection. That's when your cells are first infected with the virus. So we see some symptoms at the very beginning when your cells are just starting to become infected. Okay, and what you see here with the green line, this is the amount of virus that's present in your blood. The orange line here, this is the amount of antibodies that your body is producing against the virus. So in this initial infection, just like we see with primary immune response, it takes a while for the antibodies to start to be produced, but then antibodies are produced in a higher amount, and as the antibodies are produced, the number of virus go down. This point right here, several years later, where virus is at a low level, this is the latent period. And it may look like your immune system has the virus under control, but what's really happened is that virus has inserted its DNA into yours. And it's just kind of waiting for a signal, waiting for the time to start producing virus again. So after a number of years, it could be several years to even a decade, the virus becomes active again, starts producing its mRNA, producing more of itself, and as it goes up in number, your immune system, which is in purple here, starts to go down. This is the number of CD4 T cells. These are those helper T cells that help to stimulate your immune response. goes down, down, down until you can't fight off any other infections. HIV is transmitted only directly through contact involving transfer of certain body fluids. So HIV is not easily transmitted at all. It requires direct, close contact of body fluids. The body fluid that has the most HIV present in it from an infected person is the blood. So usually a lot of times with HIV we will see blood-to-blood contact as the cause of transmission. HIV is also present in semen and in lower levels in vaginal fluid. So we also consider HIV a sexually transmitted disease because it can be transmitted in these ways. But again, it's through direct contact with these bodily fluids, usually into an open wound source or a mucous membrane. There is low levels of HIV that's detectable in the breast milk of somebody who's infected. So women that have babies, women that are HIV positive that have babies, are encouraged to not breastfeed. There is a little bit of virus that you might find in urine, tears, sweat, and saliva, but it's so minimal it's below the infectious dose. There's so little virus in it that you would have to literally come in contact with buckets of these different bodily fluids in order to cause an infection. So we don't consider them sources of infection. Alright, so these are the different ways that HIV can be transmitted, and again, there's very few. And really, it is intimate blood-to-blood, body fluid-to-blood contact. So through needles, blood exposure, doing other kinds of through other kinds of intimate contact, and then exposure through intercourse.